So let us come to the last part of this week's content, the Mendelian genetics. So this is the last chapter of this week. So moving on with Mendelian genetics, first I would like to discuss a few terms. First of all, alleles. So what are alleles basically? Allele is for every gene there is an alternative trait. So therefore we can say that one gene can have two alleles. It can be two same allele or it can be two different alleles. So when it is same allele we call it homozygous, and when it is different we call it heterozygous. So allele can be of two types, dominant allele and recessive allele. So we can see that when it is a dominant allele, that means only that phenotype which the allele is expressing, that will be expressed, and the recessive allele will be suppressed by the dominant allele. So therefore, if there is a genotype and the capital A is the dominant allele and small a is the recessive allele, then we can see that if one gamete has the dominant allele and the other gamete has the dominant allele, then it will be both a homozygous dominant. And if only one capital A and one small a is there, then obviously it will be the expression of the dominant allele only. So this kind of genotype is known as heterozygous. And if this is also the same thing, and if here only this person will have the blue eyes, so this small a is the recessive allele trait for blue eyes. So here only the recessive allele will be expressed, and wherever the recessive allele is along with the dominant allele in a heterozygous condition, then the recessive allele will never be expressed, only the dominant allele will expressed. So this genotype, small a, this genotype will have blue eyes and the others will all have brown eyes. So these are the few terms. So allele is alternative form of a given trait or a given character. So suppose for eye color, one allele, dominant allele is the brown color and the recessive allele is the blue color. So whenever the dominant allele a capital A is present, then obviously everyone will express brown color. But only when the two alleles are of the recessive type, small a, then only we will have an expression of the blue color. So one alternative pair form of a given pair, tall and dwarf, these two can be a contrasting pair of characters. So this can be tall, can be a dominant allele, and dwarf can be a recessive allele. More than two alleles can exist for a specific gene, but only two of them will be found within any individual. So only we can have two alleles for one particular trait or one particular character. Now allelic pair is the combination of these two alleles like I was talking about, and homozygous is obviously when two alleles are same type, like capital D or small d. This is a homozygous recessive and this is a homozygous dominant. So two alleles are of the same type, then it is called a homozygote. It is called pure line. And heterozygote, when each individual contains one member of a gene pair, then means that is capital D, small d, then one allele of each type is there in that combination. So this is called a heterozygote. Now what is genotype? Genotype is a specific allelic combination for this set of genes. That means this DD or capital D small d or capital D small d, this is known as the genotype, which is the combination of genes, allelic combination of a particular gene is known as genotype. And what is the phenotype? Phenotype is whatever outward or external expression is there according to that genotype, we call it phenotype, or the physical appearance. Suppose the genotype is capital D small d, but the phenotype is only dominant. Suppose the genotype is small d, the phenotype is recessive. Suppose the genotype is capital D, the phenotype will be dominant. So this is a genotype and this is phenotype. Dominant is that allele which can express itself at all times. Whenever it is present in a homozygote or it is present in a heterozygote, it will always express itself. That is called a dominant allele, and it will suppress the character or expression of the recessive allele. So recessive allele, of course, the allele whose expression is suppressed by the dominant allele, that is the recessive allele. What is a pure line? A population that breeds true for a particular trait. That means the pure line is everything in homozygote condition that will be considered a pure line. Heterozygote is not considered a pure line. Now coming to backcross and test cross. For that, let us first understand this phenotype and genotype. So one chromosome with this allele A, capital A, and the other chromosome with small a, these two are homozygous, capital A. This is heterozygous, capital A, small a, and this is homozygous, small a, small a. So now coming to the phenotype and genotype, if it is homozygous, then it will obviously show this purple color. If it is heterozygous, then it will show this purple color. If it is heterozygous again, it will show this purple color. And if it is white, then only when small p is there, homozygous condition, then it will show white. So genotype is of three types. 1:2:1. And phenotype will be of only two types, 3:1. So this is the difference between physical appearance, phenotype, and the genetic combination, genotype. Now, how are these ratios calculated? So, let us first come to this. First, let us start with this one. If in the parental generation we have two chromosomes like this, then they will form gametes containing capital A, and this will form gametes containing small a. It might be male or female, and this also might be male or female, vice versa. So now if coming from this male parent, this capital A gamete will combine with the small a gamete of the female parent. So these two in the first generation, first filial generation or F1 generation, these two will combine and form homozygous AA, heterozygous AA, and homozygous small aa. So these four combinations are possible with gametes coming from these two individuals of the parental generation. So all these four gametes are showing three kinds of genotypes, capital AA, capital A small a, and small aa. And the phenotypes are only of two types, 3:1. So here in the F1 generation of a monohybrid cross, that is a cross involving only one pair of allele or only one set of genes, that cross will be, the ratio of phenotype will be 3:1 and the genotypic ratio will be 1:2:1. So these two ratios you must keep in mind. The phenotypic and genotypic ratio are always different. Now coming to what is a test cross and what is a back cross. So test cross is a cross, like the name suggests, that whenever we do not know what is a genotype of a particular species, it is an unknown genotype and we are trying to find out what is the exact genotype of that organism. Therefore, we are doing this test cross. So how can we understand that? So a test cross, where suppose we have a capital Y and small y. So this kind of genotype will obviously express the dominant trait of the capital Y. So whenever it is, suppose Y denotes yellow in color and small y means green in color. So if it is capital Y, it will obviously show yellow color. If it is capital Y, it will obviously show yellow color. Only if it is small y, it will show green color. So we now want to understand what is the genotype of this yellow colored thing. Is it capital Y, or is it capital Y, small y, heterozygous? So to understand whether it is homozygous yellow or heterozygous yellow, what are we doing? We are doing a test cross. So what is a test cross? Test cross is a cross between this unknown genotype and the recessive parent. So we don't know the unknown genotype, but we know there has to be a dominant allele, capital Y. Otherwise it will not show the yellow color. So we can say a capital Y and a question mark are the two gametes we are getting. And we are crossing this with the recessive parent, which is small y, the green color. Now we know that from here we can only get one type of gamete, that is small y. And from here we can either get two types of gamete or we can only get one type of gamete according to whether it is heterozygous or homozygous. Suppose if it is homozygous, then we will only get one type of gamete, which is capital Y. So capital Y coming from here and small y coming from here, these two will result in all yellow colored F1 generation. So everywhere this capital Y will go, and therefore the dominant expression will be yellow in all the offsprings. So all dominant offsprings indicate that the unknown genotype is a homozygous dominant genotype. That means capital Y, capital Y. Now suppose if it is a heterozygous, then we will have two types of gamete from here. Capital Y, small y, and the recessive parent will only have one type of gamete, that is small y. So capital Y, small y will give this heterozygous thing. And if it is small y, small y, small y will give again green color. And this will be repeated. So our 1:1 ratio will be there of yellow and green. So then we will understand that the unknown genotype is a heterozygous genotype. That is not a homozygous dominant. So in this way, by means of a test cross, by means of a recessive parent, we can understand by the cross and the results of the offspring that whether the unknown genotype is heterozygous dominant or homozygous dominant. Now, so therefore the test cross has to determine the genotype of the dominant phenotype, and also the test cross is a cross between the dominant phenotype and the recessive phenotype, and we are trying to understand what is the genotype of what is the unknown genotype. Back cross is the cross when F1 hybrid is bred with any of the parent. So suppose this is the F1 hybrid, like I have already said, if it is homozygous tall, then it is capital T, capital T. If it is homozygous dwarf, then it is small t, small t. So here only one type of gamete will come, capital T. Here only one type will come, small t. Now if these two gametes are come together in the next generation, then it will be capital T, small t, all heterozygous tall. 100% heterozygous tall plants will be here. So if 100% heterozygous tall is here and then we cross it with a recessive parent or any of the parent, whether it is the recessive parent or the dominant parent, then we will get a cross something like this. If it is with the recessive parent, we will get a cross like this, and just like here in the F2 generation, we will get 50% heterozygous dominant and 50% recessive type. So therefore we will this back cross helps to recover useful characteristics in the parental population. So from here, from the back cross, we can get offsprings with a heterozygous combination. Also we can get offsprings, we can restore the population of this dwarf or the recessive parent. So this is a back cross. So as you can understand from here that every test cross is a back cross because we are crossing it with the recessive parent. But every back cross is not a test cross because here we know it is a heterozygous tall and then we are crossing it with any of the parent. So it is not necessarily a test cross, but in a test cross, all test crosses are back crosses. So now coming to this monohybrid and dihybrid cross. So from up to here, back cross and test cross, I think it is clear that the cross of an F1 hybrid to one of the homozygous parents is the back cross, and the test cross is the cross of any individual to a homozygous parent is used to determine whether this individual is homozygous dominant or heterozygous. Then we call it a test cross. Now coming to a monohybrid cross and a dihybrid cross. Monohybrid cross, like I was saying, that a cross between parents that differ at a single gene pair. So only one single gene variation is involved, then we call it a monohybrid cross. So it is a cross between two pure organisms in order to study the inheritance of a single pair of contrasting character. And it is the cross between two organisms in order to study the inheritance of two pair of contrasting characters. So here if you can see that both round both round type character and yellow green character, both contrasting characters are used here in the cross. So this kind of a cross is known as a dihybrid cross. And here only one character, the seed color yellow and green, this was used in a cross, so therefore it is a monohybrid cross. So in monohybrid cross, like I have already discussed, the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. But in dihybrid cross, the ratio of phenotypic ratio will be 9:3:3:1 and the genotypic ratio is 1:2:1:2:4:2:1:2:1. So this is a complicated. In the F2 generation, the dihybrid cross gives a complicated genotypic ratio. So let us now understand how exactly this phenotypic ratio is coming. This is easier, 9:3:1. So this kind of a combination or checkerboard can be formed using two of these parental generation parents. So here if you can see, it is a dominant parent, capital Y and capital R only, and here it is a recessive parent, small y small r. So in the F1 generation, obviously the phenotype will be round and yellow both because of these dominant characters. So in F1 generation we can only get heterozygous dominant. Now, if these heterozygous dominant are giving off gametes, so these kinds of gametes will be there. And if it is self-crossed, then with another heterozygous parent, then it will have all the same kinds of gametes. Capital Y, capital R, so these are the possible combinations. Small y, capital R, capital Y, small r, small y, small r. So these four combinations, if combined with all these four combinations, they will give rise to these kinds of phenotypes. So now if you can see, wherever there is small r present, the seed is not round. It is not a proper round shape. I can say it's irregular shape. And wherever there is small y, you can see that it is green color. So this kind of phenotype, if you calculate the number of round, it will be 1, 2, 3, 4, 5, 6, 7, 8, 9. 9. So these 9 comes here, round yellow. 3 is green yellow, 1, 2, 3, and 1 is yellow but not round. So this is 1 over here, and here it is green and not round. So it is 1 over here. 3, 1, 2, 3 and 1 over here. So here is the ratio of 9:3:3:1 of phenotypic ratio in a dihybrid cross. So I hope this is clear. Now coming to the Mendelian laws. So Mendel's law of inheritance is of three types. The law of segregation, saying that during gamete formation, the alleles of each gene segregate. Like I was saying, the gametes can be formed of two types. From heterozygous parent, it can have two types of gametes. So why? Because the alleles will segregate from each other and it will go into separate gametes during gamete formation. So this is the law of segregation. So the alleles will separate and go into different gametes, and then it will combine independently. So that is known as law of independent assortment. Now the traits or alleles will segregate independently, and therefore they will combine and form different combinations of gametes or different combinations of individuals in the next generation. This is known as law of independent assortment. And the law of dominance says that when two alleles are together, one will always be dominant and the other is recessive. Therefore, when they both express, only the dominant allele will have the phenotypic expression. The other allele will be recessive, and it will be it will be not expressed in the heterozygous condition. So this is known as law of dominance. So these laws were given by Mendel, and these are known as laws of inheritance. And Mendel, he himself observed 7 traits in pea plants, and he observed that they had alternative characters, like flower color is either purple or white, seed color is either yellow or green, seed shape is either round or wrinkled, like I had already discussed in the dihybrid cross thing. So these are the characters which have alternative combinations or alternative characters, and therefore Mendel used these traits to perform his experiments, and it was easier for him to understand which one is dominant, which one is recessive, what is the combination, and how exactly these colors or these shapes are coming. So from here he formulated these laws, and these laws are known as Mendel's laws of inheritance. Now, coming over here, so Mendelian inheritance, Mendelian inheritance is basically the inheritance in which, or the Mendelian trait that is controlled by one single locus in an inheritance pattern. So locus is a particular location of an allele in a gene or in a chromosome. That is known as a locus. It's like a map. So in a chromosome, at each locus, one allele of a gene pair is there. So one gene has one pair of alleles. So in 2 chromosomes, if here is one allele, in other chromosome, the other allele will be here in the same locus of both of them. So they will form a pair. So if capital A is here, small a will be here, and then they will form a pair. So this kind of the genes or the characters which are expressed by this is known as Mendelian trait. Now, coming to non-Mendelian inheritance. So some phenomena have occurred which show that Mendel's laws are not valid. How they if we come to this example, you will understand better that this is an example of incomplete dominance. So incomplete dominance, this flower called Mirabilis jalapa is a 4 o'clock flower. Every day at 4 o'clock this flower blooms. So this experiment was performed, and it was seen that 2 4 o'clock plants show a common exception. So this is an exception that some alleles, the alleles of flower color, like there is a red flower, capital RR, and there is a white flower, WW. So this white flower and red flower, when they are crossed together, they form pink flowers, so which is a new phenotype completely. Now, in order to understand this, you have to understand that the law of dominance is not working here. Because if it is working here, then either we will get red plant or we will get white plant, and we will understand which one is dominant and which one is recessive. But if we get this kind of an expression, incomplete dominance, that is a pink flower, that means that the phenotype is pink because both the alleles, the white allele as well as the red allele, are not completely dominant over the other. Therefore, they are showing a mixed phenotype, and this is known as incomplete dominance, which is given by capital R small r, and this heterozygous condition is an example of incomplete dominance. So here, if you see, we have all pink flowers, and now when we cross them in the next generation, if you can see here in the F2 generation, again the parental phenotypes are coming back. 2 are pink and 1 is red and 1 is white. So 1:2:1 ratio is again coming back over here. So this is a very good example of non-Mendelian inheritance, because Mendel's law of dominance does not work over here. Another thing is codominance. So here, if you see codominance, you will see that in red and white color, both colors are expressed in the F1 generation. If it is self-fertilized, if another F1 plant is crossed with it, we will get back these two again. So here the color is not pink. That means that both are showing dominance. Here both red color and both white color are expressed separately, not mixed together as pink, but they are expressed separately, which means that both these alleles, the red allele and the white allele, both these alleles are dominant over each other and they are expressing itself individually. So this kind of phenomena is known as codominance, and it is also again a non-Mendelian inheritance pattern. This kind of codominance can also be seen in our blood group. We have the ABO blood group system. This ABO blood group system is because we have three alleles. So this is known as multiple allelism. This is another very interesting feature, that our blood group alleles are 3 in number. The A, the B and the O. 3 alleles are there. Now, any two can combine with each other and form a different blood group. So this is not a matter of 2 alleles only. So far we have seen that only Mendel said that there are 2 alleles of one single character or single trait, and therefore there is 2 alternative contrasting characters. But here we see there is not only 2 but 3 contrasting characters, and from here we have 4 different blood groups: A, B, AB and O. So how are they codominant? First of all, we have to understand that whenever A and B are together, we see codominance. We have a blood group called AB. But if one parent gives the A allele and the other parent gives the O allele, then A is obviously dominant over O, and therefore it will phenotypically show the A blood group. Same goes for A, and same goes for B and O. So if one parent gives the B allele, the other one gives the O allele, it will show BO genotype, but the phenotype will be as blood group B. And only if the recessive allele O comes from both the parents, we get the O blood group. So B blood group and A blood group can be both heterozygous or homozygous, and only AB is the codominance is shown over here, and O is the recessive allele. Homozygous recessive is O. So both A and B allele are dominant over the O allele, which is recessive, and only when A allele and B allele are expressed together we get the codominance, we get to see the codominance. So this is a codominance, and this is multiple allelism also. This is a very interesting example of non-Mendelian inheritance. Here. So difference between incomplete dominance and codominance, I think it is clear to you. In incomplete dominance, both alleles will express each other, and it will not form something called it will be completely pink, that is a mix of the 2 characters. But in codominance we will see colors like this. Both white and red colors will be seen here. This is known as codominance because this is not a separate color. This is not pink or any other color, but this is both red color and white color expressed side by side together. So this is codominance. Both are showing dominance, and here it is incomplete dominance. None of them are showing complete dominance. So that's why it's called incomplete dominance. So these are 2 non-Mendelian inheritance patterns. Now, coming to the next one is the polygenic traits. That means these traits are controlled by more than one gene. Not only one gene, but this skin color is contributed by many genes. Therefore, the height, people can be short or tall or anything in between. Some people can be very tall, some people can be very short, or it can be any height in between. Or skin color also. Some people will have a very dark tone, some people will have a very white tone, and there will be people who are in between. So these genes, these traits are controlled by multiple genes. So this allows a variation, a great variation from person to person. Each gene plays a small role in the expression of these traits. So if you can see, there are a number of phenotypes and frequency of the phenotype is given like this. So some will be at this end, like some will have a very short height and some will have a very tall height and some will be in the middle of average height. And every step there is some people who will show that range of height. So we can understand that there are multiple genes, the more than one gene are controlling these traits. Therefore, we get a lot of variations in these particular characters. Now, before coming to linkage, so let us understand that these important terms, codominance, incomplete dominance, multiple alleles, ABO blood type and polygenic traits, these should be very much clear to you. And now coming to the last part, that is linkage. So linkage, as the name suggests, it is another deviation from Mendelian principles. It says that two genes, it deviates from the law of segregation and law of independent assortment. Here, in Drosophila, this experiment was performed by Morgan, and he proved that linked genes, they occur in the same chromosome. There are some particular genes which are present on the same chromosome, and they are linearly arranged along the length of the chromosome, and they do not segregate during gamete formation. They do not segregate. They do not independently assort among themselves, randomly combine with other gametes of the same or different type, and they do not do that. There are some genes which maintain to be together. They do not segregate from each other. They do not show independent assortment. And there is a tendency to maintain the parental combination of genes. Also, the strength of the linkage between the genes is inversely proportional to the distance between the two. This can be explained by here. So, if you can understand that these are two chromosomes. So, if this in these two chromosomes, if no crossing over has happened, then it will all be like this. So 4 chromatids will be like this and no crossing over has happened. So full linkage, 100% linkage is seen over here. That means that A and B are linked genes. So these linked genes will be always together. They are not separating from each other. But here, if you can see, if crossover has happened, so one portion of the blue chromatid will go over here and one portion of the red will come over here. Now these are known as recombinant chromatids and these are the linked chromatids. So here, if you see in this chromatid, these A and B are linked genes, but here in this chromatid they are not linked anymore. So this is known as recombination. So if you understand that this strength of linkage is less between these two because the distance between them is more. If A and B has very less distance between them, if suppose B is over here, then crossing over will not be able to separate these two. Therefore, the less the distance between them, the more is the linkage strength between them. So this is known as a complete linkage phenomena when the genes are closely located together and they are completely inseparable. So they are called completely linked genes. When the linked genes are widely located like here, then we call it incomplete linkage because such genes may separate at the time of crossover. But completely linked genes will not separate during crossover also. So this is known as complete linkage and this is known as incomplete linkage. And here, if all the genes which are linked together, they form a group that is known as a linkage group. Usually all the genes present at the same chromosome side by side together, linearly arranged, they are forming one linkage group. So a set of genes at different loci on the same gene that except for crossing over tend to be inherited together. If there is no crossing over, they obviously be inherited together. Then it is called a linkage group. Unlinked, so a gene present in some other chromosome will be unlinked gene for that set of linkage group. So for that linkage group, this gene which is present in another chromosome, this is an unlinked gene. So From here, I will just conclude with complete and incomplete linkage. Over here, if you see that this is the parental generation, this is a black body, purple eye Drosophila, and this is a gray body, red eye Drosophila. So black body, purple eye, this what can I say? So in here, if you see that the gametes are of one type here, because both are recessive characters, and here there are two types of gametes showing here. So in F1 generation, 50% will be of one phenotype, dominant phenotype, and 50% will be of the recessive phenotype. This is happening because these genes are completely linked. So this is an example of a complete linkage. Now here, if crossing over has happened, then it will form these gametes also. Now there are from 2 there are 4 different gamete combinations from this parent only, and therefore there will be a number of possibilities of phenotypes will also increase, and so this population will be more than 25%, and this will be always less than 25%. So the recombinant types will be less than 25%, and the parental types will be more than 25% each. So this kind of F1 ratio will be coming in case of incomplete linkage. So here, if you see that this is a dihybrid ratio, and dihybrid ratio of 3:1, this kind of linkage is complete linkage. But in case of incomplete linkage, this will not happen. The incomplete linkage will have 2 parental types more than 25%, while each of the recombinant types is less than 25%. And all the genes arranged linearly in one chromosome are usually inherited together if crossing over has not happened, and therefore they form a group called linkage group. So in humans, since we have 23 pairs of chromosomes, therefore there are 23 chromosomes. And in Drosophila, since they have 4 pairs of chromosomes, so there are 4 linkage groups. This is all for the linkage group and the complete types of linkage. So here you will see 50% of each parental type coming in F1 generation if it is complete. But if it is incomplete linkage and crossing over has occurred, then small g will come over here, capital G will come over here, and these two will again form these 2 gametes. And from here, if you understand that this crossing over percentage is very less, around 3%, therefore the recombinant types percentage will also be very less than 25% each, and the parental types will be more than 25% each. So this is all for this week's syllabus, and I hope you go through all the videos and then we can discuss about them, and if you have any queries we can answer them in the live class. So thank you.